Good day. I would like to start by thanking the conference organizers for giving me this opportunity to give this talk. The title of my talk today is Molecular Circuits for Communications and Signal Processing as Enabler of the Internet of BioNano Things. This is joint work with my research students Hamdan Awan and Usman Riaz. I would like to acknowledge their contributions. Let's begin with a subject that we are all familiar with, the Internet of Things. The Internet of Things consists of sensors, actuators, and computing elements connected by communications technologies. The Internet of Things have enabled many applications, for example, smart homes, smart cities, and smart grid, and so on. We can think of IoT as a collection of silicon-based devices that sense, actuate, compute, and communicate. The electronic information processing paradigm is certainly a powerful one, but it is only one of the many information processing paradigms. What I'm showing you here is a cover from a book written by Dennis Bray, who is a very well-known biologist. The title of the book is Wetware, and the subtitle is A Computer in Heavy Living Cells. The picture on the book cover is a bacterium. We can see a lot of molecules now, but what the author actually wants us to think about is, the author wants us to see beyond the molecules and think about the cells as an information processing engine. So how do cells process information? They use molecules and chemical reactions. In a cell, the chemistry is not only to make molecules, but also to process information. And that is the theme of this book. Now, since cells can sense, actuate, compute, and communicate, so they behave like devices. So why don't we try to use them to build devices? This is exactly what the researchers from synthetic biology, chemical engineering, medicine have done. They have engineered cells or bacteria as devices for diagnostics and therapy. Let's look at some examples. This picture shows you an example of using an engineered bacterium as a sensor. So the aim is to go and detect whether there is inflammation in the gut. Let's start from the bottom of the picture. So these are some cells in the gut. These cells, which have a darker color, denote cells that have the information. For these inflamed cells, what they will do is they will also produce these molecules, which are denoted by these red dots, and they are the disease biomarkers. Now, so what this bacterium is going to do is it's going to go and detect these biomarkers. Now, so this is a picture of the bacteria. Now, it is obviously out of scale, too big compared with the cells, but it should be a lot tinier. But it is drawn so big so that we can actually see what's happening. So some of these biomarkers, if they get into this bacterium, what you will do is you will combine with some of the molecules inside the bacterium, and in the end, you will go through a sequence of chemical reactions. And after going through this sequence of reactions, the ultimate product is, if they are these biomarkers, you will produce a molecule that we will refer to as the reporter. So the reporter will be able to say, yes, they are these biomarkers, and so the gut is, has inflammatory problems. If there are no biomarkers, then the gut doesn't have that problem. So this is an example of using cells or a bacterium as sensors. And one thing that you want to notice, there are a number of, by using a chain of reactions inside the bacterium, the disease biomarker has turned that into a reporter. Now, so the second example that I want to show you is engineered cells for diagnostics and therapeutics. So in this example, I want to show you a more complicated type of chemical reactions or more complicated computations that can be carried out by these cells. Now, so a cells may have a certain disease if two specific biomarkers are present. So in that case, what it means is you will need to go and compute a logical AND to say both biomarkers are present, and so there is a problem with the cell. And again, to carry out this logic and computation, you will need a sequence of chemical reactions to go and do that. Now, then what are BioNano things? So what I've just covered, I've shown you just now, the engineered bacteria, the engineered cells, they can be counted as examples of BioNano things. So when it comes to the term things, we can think about devices that sense, actuate, compute, and communicate. How about the BioNano part? Then, in this case, we are thinking about bacterial cells that have been modified. And if I just go back to say that for this bacteria, it's not a natural bacteria that we find in the wild. It's a bacteria that the researchers have modified so that these chemical reactions can take place. These molecules are inside the cells for this specific purpose of detecting these particular disease biomarkers. Now, the BioNano can also be referred to that we are using materials that we commonly find in biology, for example, protein and genes, to go and make these devices. Now, there are many applications of these BioNano things. Given this is a conference on BodyNet, then researchers in this community are especially interested in applications that can be done inside the human bodies. And so these BioNano things can be for cell-based therapy or for in-body diagnostics. A very well-known example is the CAR T cell, which is used for cancer immunotherapy. Now, getting these bacterial engineered cells to go and work is no easy task. There are many problems to overcome. Robustness, the genes will evolve, and you need to make sure that things work well. Now, I also want to point out that there are many other applications of these in BioNano things. They can be used in biomanufacturing, for example. Now, what I want to say is the two examples that I've shown you earlier, both of them use gene circuits. What the researchers have started looking at is to come up with protein circuits that can also be used for the same similar purpose. Now we know what BioNano things are. Then what are the Internet of Bio things? What we want to do is we want to add the ability to communicate to these devices so that they can communicate among themselves and they can also communicate with the Internet. Now, so we have defined the Internet of BioNano things. How can we make these devices successful? Perhaps we can learn something from the electronic processing paradigm. There are many great concepts behind the electronic processing paradigm. For example, modularity, scalable design, layered architecture, and so on. In particular, the electronic circuits are one of the fundamental building blocks and of the electronic processing paradigm. And our ability to go and design these electronic circuits automatically has given rise to computers, smartphones, and internet, and so on. So electronic circuits play a very central role in electronic processing paradigm. Now, what we saw earlier in our examples on nanobio things, and we saw that these sensing and decision circuits are made from chemical reactions. So what we will do is we will use this term that we call molecular circuits to go and refer to a network of chemical reactions for processing information. These molecular circuits will form the basic building blocks for the Internet of BioNano things. And we can model these molecular circuits by using ordinary differential equations because they are built from chemical reactions. Now, because they can be modeled by ordinary differential equations, one way to think about these molecular circuits is that you can think of them as analog computers. So in this case, the concentration of a chemical species can be interpreted or can be used to go and encode a real number. And that is the same as the analog electronic circuits that you have seen before. Use voltage to go and encode a real number. Now, we use electronic circuits to go and define Boolean logic circuits. So we can do the same with molecular circuits. In this case, logic 1 may mean that the concentration of a certain species is above a threshold, otherwise it's 0. And just in the same fashion that we have used for the electronic circuits, voltage above threshold is 1, otherwise is 0. Now, so what work has been done on designing these molecular circuits? Now, if I talk about circuits that use the genes as one of the components and using Boolean logic, then this is actually the most advanced part. There are CAD tools that you can use to go and design these circuits. Now, for digital circuits, what I want to point out is there is a very interesting energy accuracy trade-off. So let's look at the graph at the bottom here. On the horizontal axis, I have computation precision. On the vertical axis, I have power consumption. There are two curves here. One is for digital, one is for analog. So what it says is if the precision that we need is just 1 or 2 bits, then an analog circuit is going to consume a lot less energy compared with our digital circuit. Now, this graph is also very similar. On the horizontal axis, again, computation precision. On the vertical axis, I have the number of molecules. And so it behaves in the same way. Now, so we will come and talk about this sort of trade-off again later on. But the important thing that I want to point out to you is if the precision that you need is small, then analog circuits are a lot more efficient than a digital one. Now, so in computer science, there's quite a lot of work on using chemical reactions as a generic analog computer. In this case, there are actually some very generic results. Perhaps one of them is there are actually methods to go and realize any given linear time invariant ordinary differential equations by using chemical reactions. Okay, these are very powerful results because they can create generic programmable computers on the chemical substrate. However, this universality comes at a price of complexity because this implementation needs a lot more chemical species, molecular species, and reactions in order to achieve this general purpose computers. So this argument is something that you probably have heard of when people go and compare a CPU and ASIC. An ASIC is more complex to design, it can be faster, but of course it's not as general as a CPU. So the same type of trade-off appears here. Now, when it comes to BioNano things, because we only want to put it into a cell, we don't want to overburden it. So my position is that for BioNano things, we should try to use a low complexity circuit. Now, so let's start with thinking about molecular circuits for BioNano things. So later on, what I will do is I will show you some of the results that we have got. What we have done is we have designed molecular circuits for two purposes. One is we have designed molecular circuits to go and demodulate molecular communication signals. Another one is we have done it to solve detection problems for signal processing. When it comes to our work, we use analog circuit is 1 bit because that will give us a low energy, low capacity design. In our work, we think about accuracy, but we also want to think about circuit complexity. So these are the focus of our work. Now, you may ask, isn't 1 bit boring? Now, when it comes to a silicon substrate, yes, it is. We know what to do with 1 bit. But when it comes to a chemical substrate, it's not that clear. It's not necessary that boring, because I will tell you an open problem in biology. So if you look at a fruit fly embryo, there are many nuclei. And these nuclei can in a very short time tell whether the concentration level of a very particular chemical is above or below a threshold. Now, what's the molecular circuit that can do that? Now, when it comes to the biologist, there are proposals, but there are no definite answers. So this is still an unknown territory. So what I see is, for certain problems, even if it's an analog 1 bit detection problem, then it is still a very interesting problem, even in biology and as well as in engineering. Now, so before we go and describe the detection problem, we will first explain what signals can actually be produced by chemical reactions. So what I want to show you here is that chemical reactions can actually produce a very rich set of dynamic signals. Now, we know from biology that cells can actually produce very different temporal concentration profiles, so varying concentration of a certain chemical species. Now, so this is an example. And so when this molecule is combined with this molecule will go through some chemical reactions. Now, so there are two sets of chemical reactions here. They share some common elements, and so this ERK, which is the name of a molecule, and is common in these two sets of chemical reactions, or these two molecular circuits. Now, but in this reaction, the concentration profile of this ERK over time is different from the other circuit. So this is time axis, and on the vertical axis, this is the concentration of these ERK molecules. So for this particular circuit, it will produce a transient behavior of the number of ERK molecules over time. So it rises over time and it drops down quite quickly. But when it comes to this circuit, the level of ERK stays up for a longer time. So what I want to show you is, by using different chemical reactions, we can actually obtain different time profiles of these molecules. So one thing you can think about chemical reactions as is they are there as generators of time profiles or dynamical signals. And that's one way to go and think about chemical reactions. Now, so what I want to present to you now is an input-output view of what a detection molecular circuit may look like. And so the problem that we want to consider is to go and distinguish between two chemical reactions. Now, let's focus on, say, we do build molecular circuits that allow us to go and distinguish between two chemical reactions. What will it look like? Now, so one point of view is we think about these circuits that from an input and output point of view. Now, you can think about that there are some input molecules. When they are present for this particular circuit, what it will do is it will go and produce some output molecules, which I have drawn as these orange circles here. Now, so this is sort of from a chemical reaction point of view. Input molecules going through a circuit will produce the output molecules. How about from a signal processing point of view? Now, we have two sets of chemical reactions that we want to go and distinguish. So the first one I call chemical reactions set 1. And for this set of circuits, as we said earlier, chemical reactions can be used to produce dynamical signals. So it produces a signal which has a low number of or low rate of producing these input molecules. Now, and then there is the other signal, the other situation that we want to distinguish it from, and this is the chemical reaction set 1. Here, this set of chemical reactions will produce these input molecules at a higher rate. So what you see here is the level is higher. And the aim is we want to go and distinguish between these two signal molecules. So because what we want to do is we want to go and do detection. We want to distinguish between these signals. So one requirement that we may want to impose on our molecular circuit for doing this detection work is, if this chemical reaction is present, then this molecular circuit will produce a small number of output molecules. If this set of reaction is present, then what you will do is this circuit will produce a larger number of output molecules. So what that means is, from the number of output molecules that this circuit will produce, we can infer that whether reaction 0 was used or reaction 1 was used. So that's an example of detection. Now, the view that I've given on the last slide is a very simplified one. Since a molecular circuit is actually a dynamical system, we want this detector circuit to work in a dynamic way. Now, so let's consider the computation of a log likelihood ratio. So I have here the probability that the observations in a certain time intervals. So these observations here is, so you can think about the observations that your company is seeing in detection. The observations are observed in a time interval generated by reaction 1, to the probability that observations in the same time interval generated by reaction 0. Now, so if you take log, then that becomes the log likelihood ratio. Now, and so if you go back to think about the standard way that we define a detection problem, then we think about, we have two unknown time profiles, two unknown dynamical signals that we want to distinguish, and one of them is applied, and then we get the observations. Now, by using these observations and the probability distribution that we assume, we can go and compute this log likelihood ratio. And what I'm showing you here is maybe the log likelihood ratio is going to be calculated. Maybe the log likelihood ratio appears this form. This is time, and that is the log likelihood ratio. Now, so what do we want to use our molecular circuits for? So now, these time profiles that we think about, they can be generated by chemical reactions, because this is what I'm showing you earlier. So we can think about that is the way these time profiles are now generated by chemical reactions. So I have two sets of chemical reactions. Time profile 0 is generated by chemical reactions at 0, and so on. Now, so what it means now is, if I use one of these sets of chemical reactions, an unknown one, then it will produce a certain time profile of the number of input molecules over time. Now, these input molecules will react with the molecules inside this detection circuit, and they will produce output molecules. Now, and how the number of molecules over time are we denoted by YT. So if this is a plot of, say, the number of output molecules over time produced by these molecular circuits. Now, if we can make this output YT to be proportional to LT, then what it means is this molecular circuit can be used to go and compute log likelihood ratio, or to go and compute probabilistic quantity. And this is what we want our molecular circuits to be able to do, to go and compute these probabilistic quantities so that we can go and distinguish between different signals. Now, we can extend that to the case of molecular communications. So in this case, what we have is we are going to have two devices. One of them we call the transmitter, and the other one we will call is the receiver. Now, what the transmitter will do is it will have these reactions that will produce these signaling molecules. Now, so we can think about these reactions as those reactions that will produce the time profiles for us. So in the jargon of communications, one dynamic signal, we just call that a symbol, and so there are two symbols, symbols 1 and 2. Now, so once these molecules have been produced by the transmitters, they will get diffused in this fluid medium. Now, some of these will come to the receivers. At the receivers, what we have is we have these molecules that we call the receptors. Some of the molecules if a single molecule has bound to a receptor, then we call it a bound receptor, and it can produce certain effects within the circuits, then we know. Now, some of these receptors are not bound. So, and what we in biology a lot of times is when these receptors are bound, they will produce certain effects within the cells. And so we can use these bound receptors as an information as to what the outside looks like. And that's the idea of doing molecular communications. Now, so when it comes to signal processing problem, what the receivers wants to know is whether the transmitter has sent symbol 0 or symbol 1. So this is, as you know, can also be formulated as a signal detection problem. The only difference is now the transmitter and receiver are spatially separated, and there is a medium that's separating them. It's just the spatial separation. But when it comes to information processing problems, that's exactly the same as a detection problem. Now, so let's see how we are going to formulate this detection problem. And so I have redrawn the pictures that from the last slide. So I have the transmitter, and then I have also the receiver. What I've done is I have divided the receiver into two parts that I call the front end and back end. The front end will only have the receptors, and I will come to what the back end is later on. Now, so I have a signal that I call x star t, and that's the number of bound receptors at time t. Now, and I'm going to define a history of x star t, all this, the number of bound signals over time. And so I consider the collection of all the trajectory of the number of bound receptors over time, and I call it using this script x. Now, so I don't know which symbol has been sent, so I have pure hypothesis, and hypothesis HI is that symbol I has been sent. Now, I can go and do formulate in a probabilistic manner. I can use likelihood ratio, and that's the probability that I see this trajectory x star given hypothesis x1 divided by the probability I see the same trajectory given the hypothesis x0, and then I log it, so I get log likelihood ratio. But I can also just consider the log likelihood Li, and in this case I look at just the likelihood of seeing this trajectory given the hypothesis HI. Now, so I can either compare LT and LIT, the theory is very similar. Now, in order for us to go and solve this detection problem, we will need to go ahead and have a stochastic model that can model both the reactions here, because this is the bounding of the signaling molecules to receptor. There's also reactions and also diffusion, and perhaps inside this medium there may be convection and chemical reactions too. So what we have learned from biophysics is that we can go one way to go and model both chemical reactions and diffusion in a stochastic way is what is called the reaction master diffusion equation, and that is the model that we have chosen. Now, there are some alternative models, but we haven't chosen them because they may not be tractable, or they do not have as a fine granularity as the reaction diffusion master equation. Now, so what we know now is the transmitter, the receiver front end, and the medium, all this together can be modeled by a reaction diffusion master equation. And the reaction diffusion master equation is in fact a continuous time Markov chain. So what we can do now is we can use the Markovian theory to go and derive an ordinary differential equation to see how LT will evolve over time. Now, we have some more general results. Instead of using receptors, we can replace that by a generic network of chemical reactions in the front end, and we can still go and derive the ODE. Now, what is this back end that I've been talking about? And now, front end will produce this result, and so this x star t, which is the number of bound receptors over time. What back end will do is we envisage that this back end will go and do the computation of the log likelihood ratio or log likelihood. Now, but ultimately, we don't want to do calculations in the same way that we use silicon-based computers, and because what we can do is we can get an ordinary differential equation to see how actually evolves over time, and chemical reactions are like analog computers, and so they can go and implement ordinary differential equations. So in the end, what we want to do is we want to go and use chemical reactions or molecular circuits to go and implement this backend. Now, so what does this backend look like? We can show that this backend looks like a bank of filters, just the same as what you will see in many communications textbook. So that's the signal, the number of bound receptors over time. We can pump it into a number of demodulation filters, one filter for each symbol. And what is this L0T, L1T? They are the log likelihood of seeing the trajectory of the number of bound receptors over time, given the hypothesis that the symbol being sent is K. Now, so what does this, and then after that, once you have computed all this log likelihoods, you just choose the one that's the maximum. So it's something that you have seen a lot in communications. Now, what is the differential equation for LKT looks like? Is this particular differential equations? Now, there is a big chunk here. You need to go and differentiate the signal that we have. And now, there's this part and expectations. And what that is actually a posterior estimation of how many molecules there are near the receivers. So you can get this part by solving a Bayesian filtering problems. Now, so this is what it looks like. Now, it's quite a big chunk. Can we go and realize it using molecular circuits? Now, it doesn't look easy because this Bayesian filtering problem is actually quite a hard problem to be solved, and it's computationally demanding. Derivatives, yes, there are circuits in using chemical reactions to go and compute derivatives, but there's also a lot of work, a lot of chemical reactions are needed. We want low complexity circuits. Can we do better? No. Another problem that we need to go and overcome is that because we are talking about computing log likelihood, log probability can be positive or negative, but concentration can only be a positive quantity. Now, so there's a way to go and use concentrations or chemical reactions to go and do calculations with both positive and negative real numbers. But what you need is you need to have more chemical reactions and more chemicals to go and do that. And again, it's a complexity problem. Can we get away with that? And also the last question is, how can we implement this max operator using chemical reactions? Now, so when it comes to, this demodulation filters, I will explain to you what we have done in order to turn it into a molecular circuit. So we have actually made a lot of different choices in order to go and to be able to turn it into a molecular circuit. And first of all, for the front end, we have to try to operate in a certain region so that the rate of binding to the receptors becomes linear in the number of signaling molecules. And we also have to go and choose these transmission symbols in a particular way. So in this way, we have chosen these symbols to have a long duration, and the emission rate is the mean emission rate is a constant. Now, once we can do that, what we can do is we can then approximate the Bayesian filtering solutions by using constant. That depends on the symbols. So we bypass solving a Bayesian filtering problem. Now, also because we have used a long duration, we can go and use timescale separation. And by that, we can approximate this derivative term, and we can also approximate that by an expression x star t. So the thing is, we want a long duration, and that's essentially we want to do averaging. And so think about the law of large numbers. We want to go and average a lot of quantities, and so that is what we do in order for us to go and get good estimates. Now, the thing is, the log likelihood can be both positive and negative, and we don't want complex circuits. And so what we have chosen to do is we have chosen to round the negative log likelihood to 0. And one more thing that we have done is we are going to approximate the Hill function, and which is a rational function that's commonly used in biology to go and do this circuit. So ultimately, after all these approximation steps, then we can show that this log likelihood can be approximated by this quantity ykt, and ykt belongs to this, ykt obeys this differential equations. And this is x star t, which is the number of bound receptors, and so it's coming there, and so you see no derivatives. And there's another term that depends on the signals at the receiver that we can actually make use of, and this is the Hill function that I'm going to that I was talking about. And the thing is, we can make the log likelihood ratio to be approximately given by the output of a molecular circuit, and that's exactly what we want to do. So what I will do is I will show you some results over here now. And so there are two possibilities that we consider, whether the transmitter has sensed symbol 1 or symbol 0. So we start with saying that this, using the transmitter has sensed symbol 1. So there are two filters, filter 0 and filter 1. Now there are a few curves here. The blue dash curve here tells you the accurate log likelihood computed by using computers, and the red curve here is the one from the molecular circuits. And what you see here is the two are actually quite similar, and we do a lot of realization. We simulate it many times, and the magenta curve tells you the root mean square error. So the approximation is very good, and so our circuit can approximately compute the log likelihood quite well. Now, how about when it comes to symbol 0? Now, if symbol 0 is sent, and then when it goes through filter 0, the approximation is not as good, but it's still very well. Now, but what happens when symbol 0 is sent, but we use filter 1? The actual answer is you will have a negative log likelihood. But what we have done is we have actually rounded it, this negative log likelihood to 0. And so what you see is the output is actually very small. Obviously, the error is big, but does it affect how we do? But does it affect the result of the modulation? Because ultimately, the modulation we go and choose which filter has the higher log likelihood. Now, so if you look at if symbol 0 is sent, filter 1 is at a very high level, and the filter. So filter 0 is at a very high level, and filter 1 is at a much lower level. And so the modulation is actually correct. Similarly, you can actually check for symbol 1 is correct. So what we want to argue is, even though we have rounded this negative log likelihood to be 0, it doesn't change the modulate. It doesn't. In this case, we can still correctly to go and demodulate the signals. Now, so one thing is, this circuit that we have come up with, it can be implemented by using a gene circuit. And there is a mapping between the type of molecules. The signaling molecules becomes a transcription factor in the genes and so on. So this can be, and what we have done is we take this model and then we fit it into some gene expression data that people have obtained. And we found that our model can actually fit quite well to the existing data on gene expressions. So what it means is these filters that we have come up with can in fact be implemented by using gene circuits. Now, so what does the bit error rate look like? Now, one thing that I haven't talked about is how we implement the max operator. So what we did was we implemented using some analog chemical reactions, because max is essentially digital computations. We don't want to do digital computations because it's energy intensive. So we have chosen an analog implementation of the max. Now, so what I'm going to compare to show you here is we compare the exact the bit error rate of the exact demodulation filters, and then using the molecular circuit implementation. So what I have is I have time on the horizontal axis, and vertical axis I have the mean bit error rate. Now, as I said earlier, we are going to use the history up to a certain time to go and as the signal to do demodulation. And so if I'm going to use the signal until time t to go and do the demodulation, and so this is the t that I'm plotting. And so what it means is, as I go to the right-hand side, I'm using more data to go and do my demodulation. Now, so if I use the exact computation, then the demodulation filter does not need, and then it initially has a very high error, but when it has a lot of data, then the bit error rate becomes very small. For the molecular implementations, it stays up here for a while, and then later on it drops down. So in the end, if the time is long enough, then the molecular circuit implementation has a very good bit error rate to compare with the exact demodulation filters. Now, and one thing is, this circuit requires a longer time to go and do the computation, and filter work will try to go and reduce that. Now, so we can now decode 1 bit. Can we go and do more bits? Yes, we can extend our filter, our work to go and demodulate a sequence of symbols. One thing is we will need to go and reset our demodulation circuits, and that is as long as we can do the resetting operation, then what I'm showing you here is we can successfully decode a sequence of bits. Now, so what the next thing I want to talk about is, yes, we have done the molecular communications, we have done the demodulation problems. We can actually go, the same framework can be used for solving the detection problems. So for the detection problems, what we have is we have 2 unknown signals. It can be either generated by reaction set 0 or reaction set 1. We don't know which one of it. And then we want to have a detector to tell us which reaction was actually used. Now, so again, we have front end and back end. Now, so what we have gone on is we have gone on to go and make the front end to be a lot more complicated. And so this S is the molecules that's produced by these reactions. And then what we have is, instead of just receptors, we have X that can bind to X to produce X star. And when you have X star, we can actually go and bind to a gene. And then what we will do is we will use this signal when X star is bound to this gene to go and do this detection. So what you see here is, and so there's 2 layers. And so S is what the time profile for S changes, but we are using a lot more reactions to go and use a downstream signals to go and try to the detection. Can we do that? Yes, we can. Now, so a lot of the solutions of this problem is quite similar to what you have seen before. What we can do is we can derive an ODE for the log-likelihood ratio. That becomes quite straightforward because of the tools that we have developed. There are some difficulties for us to still overcome, and that is really to derive an approximate Bayesian filtering solutions and how to map the ODE that we have found into a molecular circuit implementation. What you found is this circuit can be implemented by what we call a coherent feed-forward loop. So what I'm showing you here is some results for the detection. And on the horizontal axis, I have the detection decision threshold. On the vertical axis, I have the true positive probabilities. There are 2 curves here. One, the blue curve here is the actual by using exact calculations on a computer. And say if we have a molecular circuit design, is an analog, what you find is the 2 methods are giving me very similar true positive rates. So what it means is the detection is actually quite accurate. So I come to the conclusions for this talk, and the key message I want to put is that an important problem to go and tackle in the Internet of BioNano things is to go and design molecular circuits. And what we have demonstrated that it's possible to go and derive these molecular circuits, and these molecular circuits to go and perform demodulation and detection with good accuracy. Now, and one thing is we purposely try to come up with these circuits so that we can demonstrate that we can actually go and use chemical reactions to go and perform these probabilistic type of calculations. Now, what I want to say is designing molecular circuits for signal processing and communication is a field is in an embryonic stage, and there is plenty yet to be done. So what I hope is that you will join us on this journey. With this, I will end my talk and thank you for your attention.